Hello everyone. So now we have seen epilepsy, right? We need to see a little bit about Parkinson, right? We are not thought about that right now, but we have discussed, right, that I will be showing it in one of the lectures how the what is Parkinson and how the current methodology to treat the Parkinson is there, and from the neural science perspective, from the experimental neurophysiology perspective, what are we doing, how we are using the electrodes to implant in rat's brain, whether it is surface or cortical implant or whether it is deep brain stimulation. So we will look into those lectures in a few days. Today the idea is to show you the surgical procedure in rodents, and in our case the rats are the animal model, and how the surgery is done, what kind of rat we use, what kind of anesthesia we use, how the surgery is done, and then how the implants are done, right. So now it is really a 100 minutes, 120 minutes of videos, right. So what I have done is I have divided those things into 4 different videos. So I will be talking about the each video before you can see that. So that will give you the gist and a base to understand what exactly we are talking about. So in the video 1 we will be looking at the local anesthesia. We will be looking at how the craniotomy is used to open the motor cortex, SMA region, somatosensory cortex, some parts, and the coordinates from red atlas. So we will just show you that coordinates, right. Let us not worry about too much worry about the coordinates right now. Particularly when you want to implant the device in a certain region, you should know the atlas, red atlas, like a brain atlas. Okay. So you understand that where exactly you need to implant the device from that atlas. In all these videos we have used rat, which is called Sprague Dowley. The age is about 3 to 4 months. The weight is around 360 grams, and we are using a general anesthesia which consists of ketamine and xyloxine. So these are the some of the constant thing. The rat breed is constant, the age group is between 3 to 4 months, the weight is almost similar, and the anesthesia technique is also similar, right. So that when we get the data from multiple rats, the constraint should not be the procedure. We can optimize the device, but let us not have lot of different exploration when it comes to surgery, right. We do not have to do lot of exploration, different methods, right, to explore and see which one is better. No, it is already known, right. So we keep it as it is. So what I have done is the first video, which is the rat surgery video 1, right. Like I said, we have 4 videos: rat surgery video 1, 2, 3 and 4. The first video, for that let me show you the slide, and then you can see the video. So the module is divided into 3 or 4 different sessions, and the module is on neurosurgery of RET for MEA implantation. As all of us now know, what is MEA? MEA stands for micro electrode array, right. Now, you need to use this electrode and to implant in that brain. That means, you have to do a craniotomy. Craniotomy is removing the part of the skull, or it is so that you can implant the device. So, if you see the slide here, you can what you can see, there is a image of deep brain region located at approximately 7.8 millimeter from the surface. This is somewhere all the way down, okay, all the way down. But here we do not have to worry about it. This is when we are looking at the Parkinson, because for Parkinson, right, Parkinson disease, the way to treat it is by using deep brain stimulation. Right, deep brain stimulation. That means, you have to go deep down in the brain and then stimulate a certain area. We will discuss that later, but this is how the actually, if you just go and understand that how the brain at last looks like, right, it is not so easy to understand, for at least for the engineering perspective, right. It will take some time for us to understand which area is what. But on a bigger picture, if you want to understand what happens when we apply electrical stimulation on the surface versus when you apply the electrical stimulation deep down in the brain, then and then effect of that, right, on the motor area. So, this is the rat's brain, representative image of the for the intended brain region, and the red color circle that you can see, which is this one, right, this one is the area of our interest, here one and here second one, okay. So, I will tell you how we are using it, and we need to implant the device deep in the brain in these two regions. This is surface plus deep, okay, surface and this one are deep, okay, for applying electrical stimulation. Now, to do that, what we have done is that we have first shown in the video, right, what are the local anesthesia technique, and we use something called lignocaine for the local anesthesia. Then we will, in the same video, you will be able to observe what is how the craniotomy is done and how the motor cortex, SMA and then somatosensory cortex, as well as the when you open, how the things are done, right. When you have to first remove the hair, and then you have to put the ethanol wipe through ethanol wipes, and then and of course anesthesia is given, and then you have to how the skin is removed, how the skull is removed, right, and how the dura is opened. All these things would be a part of it, but right now we are just showing the skull removal in the video one. So, I hope that you would like the video. If you have any questions, you can always ask. Just see the complete module, that means video 1, 2, 3 and 4, and then try to be curious and start asking questions, alright. So, let us play the video now.